I think Ruchi and Ronan did a great job setting up, the workbench and a lot of the struggles I've personally experienced as well using all of us. I wanted to go over a use case, kind of just a very simple organization of the data, how to find data, and then how to access it and try to use it. So the stuff I'll be showing is you would need control tier access. So like Ronan mentioned, you'd have to complete an application and get access to the data. But once you do, these are some of the things you can do with the data. And because last week there was a question about ClinVar variants and how to kind of look at what ClinVar variants individuals have, and with their phenotype data, I thought I would show something related to that. So starting off, this is what your home page when you log on to the workspace looks like. To click on a workspace, you just click on it, and then if you go to the analysis tab, you'll see all your active Jupyter notebooks that you can use. Like Ronan mentioned, this is where you would get a Jupyter environment, and you can specify what resources you want here. So I have one running already. So to start off, some of the things I want to how to use the user support hub to find information about the type of genomic data you're trying to use. So Ronan talked about CRAM files, but there's a lot of different kinds of data and different deliverable sets that you can access. The concept of buckets versus the persistent disk and using GSUtil, how to download the data from the Google Cloud buckets to work with, and how you can then, some of the results you produce, how you can save them to your bucket to then have later for future use. And then last, the small use case of how to combine ClinVar data with some phenotype data. So to start off with the user support hub, so if you go to your side panel, and right here is the user support hub, this is a wealth of information. So I really like this page because it has a lot of information in one place. So specifically, if you want to look at the genomic data, you go with working with the data. It kind of progresses. Once you get familiar with the workbench and watch some of the intro videos, you can then dive into the data. So specifically, if you want to look for information on the genomic data, you go to genomics, how the are organized. So if you look through this page, it gives you a list of the different types of data, CRAM files, variant data, some associated auxiliary files. So these are the 3 different classes of data that you have access to. And then it gives you a description of what the different formats are for these deliverables. And so if you go to the next page, which is the CDR directory, this is the directory that you need control tier access to look at. So essentially, this is all the genomic data. There's the whole genome data in a VDS format, which is a type of data format to store large amounts of genomic data. Then the whole genome sequencing data is delivered in this is auxiliary files, so things that you need with the whole genome data. So then we have exome data in various formats. And then ACAF is allele count, allele frequency data, which is filtered already based on an allele frequency threshold, because like Ronan kind of alluded to, it's very expensive to process the raw genomic data yourself. So what all of us has done is given you these packaged deliverables of data with certain filters that it's more manageable to use. So they're in the Hale matrix table, VCF format. They also split multi-allelic sites, Plink format to run GWAS studies, et cetera, which is really nice. So they've also subsetted the ClinVar variants into these same types of genomic data sets and formats. So I'm specifically going to talk about the VCF files, but you can also access this data as a Hill matrix table, Plink files, bgen, and a bed file format. If you want more information on these various types of formats, they're all available there on the site. So it's great to kind of, if you're not very familiar with these types of formats, it's really helpful to have this. All right, so moving on. So that's where you can find more information about what type of data you want to use. So in addition to that, if you want to look at certain ways to use all of this type of data, there are really great tutorials. So I really recommend looking through these tutorials in a lot of detail because they can be very helpful to understand how to use these different types. So they have one on using dsub, like Ronan talked about. They have a basic one on how to use the genomic data, how to process CRAMs, et cetera. So wealth of information here when you're getting started. So now the question is, where's the ClinVar data, right? So, and what type of format do we want it in? So if we go back to the information here, we know it exists. The ClinVar variant data exists in all these different formats. And so if we want to know more about, okay, so this ClinVar data in the VCF format, what exactly, what filtering has been done and what variants are in this data set. So if you want to know more about that, you can go into, so the information about the smaller call sets, and what that gives you is an idea of, yeah, so what format the data is in and what filtering has been done on the data already. So if you do get variants in the VCF format, you have to keep in mind that some of the filtering has not been performed, so you have to be mindful of that. Whereas if you use some of the other data sets, that filtering has already been performed. So this is a really nice place to find out what's been done to produce this call set, which is really important for downstream analyses. Okay, so yeah, so where is the ClinVar data? So I just showed you where that is. So now how do we access that data if we want to work with it? So I'm just going to load some Python modules, and then so like Ronan mentioned, there's a workspace bucket which you can get using this environment variable called workspace bucket. So this is essentially a Google Cloud bucket that is linked to your workspace. So every workspace has a particular bucket. However, what you also have associated with your Jupyter environment is a persistent disk. So this is essentially attached to your environment. So when you delete this environment, so not pause, but if you delete it, the associated persistent disk will also be deleted. So you have to be mindful of that, because let's say you have some files stored on this persistent disk and you want to make sure you have them even after you delete your environment. You need to copy it to your bucket for future use. So these are the two different storage areas. One is temporary, one is permanent. The difference is that you pay for storage with Google Cloud Buckets, but it's not that expensive compared to, I mean, you're paying for persistent disk storage and it's much more expensive because you're working with it in real time. So there is a thorough documentation of how to use gsutil commands, which is what you would use when you're accessing anything in Cloud Buckets and going between Cloud Buckets and persistent disk storage. Okay, so how do you access all that genomic data I talked about? So there is this environment variable called CDR storage path, which gives you essentially where the data is located in the all of us buckets. So, and to access anything any of the genomic data in the All of Us Google Cloud buckets, you need to use this Google project identify or variable, which essentially says that you have access to these files because you're in the controlled tier approved. Yeah, so essentially you can run gsutil ls to list all the files. So here's a list of all the clinvar VCF files, for example. So for this case, what I did was I downloaded a VCF file from the Google Cloud bucket that All of Us provides to my persistent disk storage to work with. And the reason why is because, like as Ronan showed you, when you work with files on the Google Cloud buckets, you have to use things like dsub to be able to request a virtual machine and then bring the files to that virtual machine and then run some commands. However, what you can also do is download those to your persistent disk and then analyze them there. So if your files aren't extremely large, like CRAM files and large whole genome sequencing files, you can download them to your persistent disk and analyze them. So in the case of these clinvar variants, they're manageable enough so you can download them and work with them. So in this case, I've only downloaded one chromosome and the smallest chromosome. So taking just chromosome 22, clinvar variants. So this essentially is a VCF file. Rows are individuals, columns are variants, and it basically tells you which individual has which clinvar variant. So I wanted to extract this information into a table where I know exactly which individual has which variant. So I used BCF tools, so I'm not going to go over that, but I used this tool to kind of extract exactly who has which variant and what genotype, whether they're heterozygous or homozygous, alternate. So anyone who has a clinvar variant, and that will give me a nice table that I can use. So anything that I'm analyzing using, like I've brought it to my persistent disk, so I'm using, I'm just accessing the files from that disk. Another benefit of analyzing data on your persistent disk, if it's a manageable amount of data, is that a lot of the genomic tools are already installed in the Jupyter environment. So you have access to them, versus if you are analyzing things with dsub and using a cloud machine, you would need to use the containers. So you need to have containers for all the tools that you need to use. So yeah, just keeping in mind. I think a lot of tools are actually already installed on a Jupyter environment, which is really nice. Okay, so now if we know who has which variant, but now we want to know more information about the variants. So there is a variant annotation table that All of Us provides. So you don't have to download this from some external resource. This table is a very comprehensive detail on every variant that exists in All of Us and information about that variant. There's about 100 fields actually in this table, what transcript it belongs to, which gene it's in, and various other. Yeah, you can scroll through this and look at it. It's a wealth of information. So the information about the variants is in this table, so we want to access that and combine it with the information that we know about the individuals and what variants they have. So just for a use case, I decided to extract variants from check2, a gene. So first I downloaded the variant annotation table to my persistent disk, and you can see it's an extremely large file, so I don't really want to read that in to my Python Jupyter environment. So instead, I extracted just variants that were for check2 and saved it to another file. And then after I created that file, so I was then ready to merge the two together. So the information about who has which variants, and then what the annotation of those variants are. So quickly, I've produced a few files, so if I do want to use these files later and I want to make sure I'm not going to lose this the data, I like to copy my files from the persistent disk onto my Google Cloud bucket. So you use the gsutil toolset to copy any files from your persistent disk back to the Google bucket. And so you can do that just like you would use bash commands for copy. A lot of the times it's gsutil, and similar to the bash commands, the source and the destination. So copy those files to the bucket so I have them for later use. Okay, so like I mentioned, that variant annotation table has about 100 columns. So I extracted all variants that were in check 2, read it in, and then I get this table with all the information on check 2 variants. What I can then do is subset that table to only include the columns of information that I want. So if I'm most interested in ClinVar classification, I will extract that, the gene symbol, of course, and then kind of get the data ready to merge with the individual level data. So essentially now I have the data set with the variants that each individual has, as well as the variant annotations for those corresponding variants. So then what I do is I merge them together to create a table. I can't show the data because I can't show individual level data, but essentially I make a new table which includes the variant, the person ID, and then the annotation associated with the variant. So the next step is to integrate phenotype information. So Ruchi went over this last time, but you can create cohorts based on certain conditions or criteria. So in this case, what I did was I created a cohort using ICD codes for diagnoses for myeloid malignancies. So specifically using certain codes I'm interested in curating the phenotypes for. So using the phenotype browser, I was able to do this. And then what I get at the end is a data frame of all the individuals with their diagnosis information. So any individual that has a diagnosis for myeloid malignancy ends up in this data frame. But that's only the individuals with myeloid malignancy. So what I have to do is make another data frame with all participants and then merge that or combine that with the myeloid malignancy diagnosis cohort to then annotate it in a column. So what I then have is one column that's the diagnosis information, and then the other columns are the information about the participants. The sex at birth is encoded in various ways. So to kind of categorize them, we tend to categorize male, female, but then all the other ones into a separate category. So 0, 1, and 2, where 0 is any ambiguous category, 1 is male, and 2 is female. And then the next thing we'd want is probably age information. There's actually some really nice resources from the How to Query All of Us Data Python tutorial. It's one of the tutorial workspaces. So they kind of give you a couple of different ways that you can calculate age, because what they do give you is the birth date and time, but not age. So one way to do that is by subtracting the current date from the birth date, and that will give you age. And so this is just the resource to that. So that's what I did. I took the current date, subtracted it from the date of birth to get the participant age. So now this is what I have, a data frame with the person ID, information, myeloid malignancy diagnosis, and age. And then I also have a table where I know I have a variant ID, the person that has that variant, and then the annotations associated with that variant. So in this case, I have the gene symbol and the ClinVar classification. So then I can combine these two again to make a massive comprehensive data frame where I have both the phenotype information of each participant as well as their variant and the annotation information. And so then what you can do is you can make different plots. So I made, for example, a pie chart to look at, so for Check2, what's the percentage of categories of ClinVar classification present in these individuals. So the ClinVar classification categories can be a bit messy, but essentially you can see majority of them are uncertain significance, and a small slice of them are pathogenic. And then you can also look at, on average, how many Check2 variants does an individual harbor. So you can plot the data. So people harbor on average 10 variants in their Check2 gene, which is interesting. And then how many variants of different clinical significance do individuals harbor? So most people harbor benign variants as expected, and then you can see on average people only have one of the other classification of variants. So yeah, this is just an example of what you can do with the data. Yeah, so I hope that was helpful.